Okay, so we're going to go ahead and pick up at section 13.3. This is slide 35. So we're going to today talk about listing characteristics of antigen that make them really good immunogenic antigens. We're going to describe how the immune system responds to alloantigens, superantigens, and allergens, which are three different classes of antigens. And we're going to list the types of cells that can act as antigen-presenting cells. Okay, so we're now into stage 2 of an immune reaction. So in the previous lecture, we talked all about B cells and T cells. And so now we've set the stage, having all of the cells necessary to produce this response. We now have an entrance of the antigen, which is actually the start of the entire adaptive immune process. So in order for the immune system to be able to recognize something as an antigen or an immunogen, a substance has to meet certain requirements in terms of foreignness, shape, size, and also accessibility. So examples of things that are immunogens, in other words, things that are going to actually start an immune response, would be whole microbes, or in some cases, their parts. So remember that a flagellum is something that would start an immune reaction. So even if you just have flagellar proteins floating around in the vicinity of an antigen-presenting cell, that may be enough, okay? Cells or substances that arise from other humans, and in some cases animals or plants or various molecules from those organisms. Complex proteins and protein-containing molecules are more immunogenic than repetitious polymers of a single type of a molecule in general. And basically it's because there are just more antigenic places. So if you have a polymer and that particular molecule isn't especially antigenic, then having lots and lots of those isn't going to be any more immunogenic than possibly having all different kinds of proteins or protein complexes that the immune system may be able to recognize. Okay, so most antigens fall into a couple of basic categories. So proteins and polypeptides actually tend to be the most immunogenic. So proteins seem to be more immunogenic than sugars of various things, but often you get the two of them together, you get a protein and a sugar as a glycoprotein together. So things like enzymes, cell surface proteins, exotoxins are examples of proteins and polypeptides. Lipoproteins, that tends to be things like cell membranes. So if you have a lysed cell, you might have lipoproteins floating around from that situation. A glycoprotein, for an example, a blood cell marker, so ABO markers are basically glycoproteins. Okay, so that's a protein with a sugar hanging on the outside of it. Nucleoproteins. So again, this is a protein with a piece of DNA attached to it. Pure DNA does not tend to be a very good immunogen. But if you add a protein to it, again, I imagine the immune system really is recognizing the protein as the antigen and then recognizing the DNA as part of that protein complex. Polysaccharides, so there are certain bacterial capsules that look very immunogenic to our immune system, but in general, proteins are more immunogenic than sugars are. And lipopolysaccharide is a really good example of something that is highly immunogenic. So LPS is actually used in a purified form as the best immunogen. So if they really want to get an immune reaction, they put something in and they add LPS to it. And the LPS really wakes up the immune system, and then it's liable to respond to the other antigen as well. Okay, so what makes a good antigen? Probably not from the perspective of our personal health, but in terms of the immune system, what is something that the immune system is going to really strongly react to? So first of all, it's the chemical composition. And Second of all, it's the context. So if you have an antigen in addition to things like cytokines, which are specifically designed to wake up portions of the immune system, that's going to increase the immunogenicity of that particular antigen. And their size actually plays a role. So things that are bigger tend to evoke a bigger immune response as well. So generally speaking, large antigens are better than small antigens, but that's not sufficient. So two examples, you have glycogen, which is a highly repetitious structure. It's like 100,000 Daltons, which is a measure of weight for molecules. And it's not immunogenic at all. So glycogen is actually a normal molecule that you would expect to see primarily in the liver, but you would see it out in the bloodstream as well. That is not going to cause an immune reaction. On the other hand, insulin, especially insulin that is not human derived, is only 6,000 Daltons. It's a fairly small thing, but it's highly immunogenic. And as we are trying to produce artificial insulin using things like E. coli and stuff like that to produce the pieces, that's something that we had to pay attention to, because especially if any part of the E. coli ended up in the soup along with the insulin, you could cause a nasty immune reaction. So that was one of those things that they really had to do quite a bit of work on when they were making artificial insulin for use in humans. Okay, so things like the things on the bottom are poor immunogens, okay? So saccharides and polypeptides that are just simply repeated tend not to be very good. But good ones are big proteins with lots of different epitopes on the outside of them. Blood is something that's highly immunogenic if it does not match our blood type. Plants, so think poison ivy, that's a really good example of a highly immunogenic, at least for those of us who really super react to any kind of poison ivy or poison oak or anything. And any piece of a microbe tends to be highly immunogenic, particularly that innate immune system is going to recognize those molecular patterns and start that whole process. And once you start killing off these bacteria or viruses and displaying pieces of it on the outside of an antigen presenting cell, that really starts off an immune reaction big time. Okay, haptens are things that are immunogenic, but by themselves, they're not quite big enough to elicit the proper immune response. So if that incomplete or too small a molecule is attached to something bigger, typically referred to as a carrier, that's enough to kind of put it over the top and make it immunogenic. Okay, so the carrier group itself is not something that elicits an immune response, but it just increases the size. And so a larger molecule with this immunogenic little thing attached to it is going to be enough to start that immune response. So the hapten serves as the epitope, and the carrier serves as the carrier part of it that will actually get it to the immune system and cause it to wake up. So here are examples of that sort of thing. So drugs, metals, ordinary chemicals, environmental chemicals, so household things. So when they combine with these larger carrier molecules like serum proteins, many of whom are explicitly there just to carry stuff around in the blood, to pick smaller things out of the blood and carry them from one point to the next. Okay, that would be enough to kind of take it over the edge to become an immunogenic molecule. Okay, so we talked about a couple of different terms that we're going to want to remember. So alloantigen is a cell surface marker or molecule that occurs in some members of the same species, but not in others. Okay, so as an example, the basis for blood grouping, the ABO, is an alloantigen. So in my case, I'm a blood type O. So I have neither A nor B on my blood cells, okay? So this is why I tend to be, why type O tends to be the universal donor, because we don't have that A or B antigen that would cause trouble in someone who is A or B. Now, on the other hand, if somebody transfuses type A blood into me, my immune system is going to go crazy, okay? Because there is an alloantigen that is immunogenic to me, okay? But if somebody with type A blood got that same transfusion, they would not respond, okay? This is also in part responsible for immunoincompatibilities that happen during organ grafts. Okay, again, something that would cause a person to recognize that tissue as not self. Okay, so whether it comes through a blood transfusion or an organ graft, it would raise an immunological response to that foreign tissue. Okay, super antigens are things that cause the immune system to way over-respond, okay? So bacterial toxins that are potent stimuli for T cells, LPS being the classic example of something that is just going to really wake up those T cells and cause an enormous immune response. And that is one of the reasons why those gram-negative organisms that release LPS are liable to cause that toxic shock syndrome. So if you are too aggressive at killing off a gram-negative infection and release all that LPS at one time, you risk causing that toxic shock situation. So, so LPS activates T cells at a rate, hundreds of times greater than an ordinary antigen. Okay, and so again, you're ripe for an overreaction. Okay, so that overwhelming release of those cytokines caused by that molecule happens, and again, toxic shock syndrome is associated with those super antigens. Okay, now we're going to be talking about allergic reactions in chapter 14, but this is the piece of the microbiological and immunological picture that actually causes that allergic reaction to happen. Okay, so we've kind of glossed over this whole concept of antigen processing and presentation. Okay, I have mentioned it. So remember we talked about macrophages eating say a bacteria. So it kind of reaches around with this little amoeba-like pseudopods and pulls that bacteria inside and then starts digesting it, okay? So it's going to identify those antigenic pieces that it needs to present to those T cells during that handshake. So it's going to take those antigens and it's going to put it up on the outside of its cell surface associated with that major histocompatibility complex, okay? That MHC molecule. So you get the MHC plus that antigen on the outside of the cell, and that's the handshake that needs to happen, okay? So antigen presenting cells are things that kind of do that professionally. They are professional phagocytes or professional antigen presenting cells. The dendritic cell is probably the biggest. That's pretty much all it does, is it goes and engulfs stuff and presents the antigens to other things. Macrophages do that as well, but they also have other jobs. And if you get a chance to look at that macrophage review paper, you're going to see some of the other things that macrophages are responsible for. So it's a little bit more of a Swiss Army knife kind of a cell, but one of the things that it does is antigen presentation. And B cells also have the ability to do that to some degree, although that's certainly not their major function. Their major function is the production of antibodies. Okay, so after processing is complete, the antigen is bound to the MHC receptor and moved to the surface of the cell. So it's readily accessible to the T lymphocytes, and that handshake can occur. Okay, so again, this is a pictorial view. So here we have a picture of a dendritic cell, and I know that because macrophages kind of look like fried eggs. They've got that nucleus in the middle, and then they have this kind of undulating thing on the outside. Dendritic cells have all those little spiky projections. They kind of look like, sea anemones. They've got these waving things that kind of help it, eat the original pathogen. And again, it's going to, as you can see, kind of coming in, there's a microbe and it's coming into the dendritic cell. The dendritic cell is wrapping its cell membrane around, it's phagocytizing, it is digesting it. Once it's in that vacuole, creates the phagolysosome, all of those digestive enzymes, and oxygen, radical oxygen species are then applied. And once the bacteria is digested, those antigenic pieces are then displayed on the outside of the antigen presenting cell with the hope that it's going to come in contact with the helper T cell to do that handshake. Okay? And the antigen that is connected to that MHC2 receptor is the processed antigen. It's just that little piece that's antigenic. Okay? And then it's going to find that helper T cell and it's going to do the handshake with the antigen that's presented on the outside of the cell. And there's a kind of a blow-up picture of what that looks like. So you have that T cell receptor that's got the two little lobes, and then it's got the two antennae, which now grab onto that antigen. Okay, so then you've got the MHC2 with the antigen now connected to that T cell receptor, which grabs onto a piece of the antigen. And then you have all those co-receptors involved. You have the CD3 and CD4 co-receptors involved in that handshake. So most antigens must be presented to a T cell first, and specifically a helper T cell, even though eventually that whole process is going to activate T cells, all of them, as well as B cells. Okay, so there are some T cell independent antigens that can trigger B cells directly without any antigen presenting cell or helper T cell involved. Okay, so things like lipopolysaccharide can wake up a B cell just by itself. Okay, um, there's a polysaccharide from the capsule of Streptococcus pneumoniae that is able to do that, that's able to bypass that whole thing. It just, the immune system gets so upset by those things being there that they just have all kinds of mechanisms to react. Um, and also rabies and Epstein-Barr virus are able to go directly to the B cells. Okay. Okay, so concept check 3, which of the following is not an antigen presenting cell? And the answer is C, the T helper cell, which is the recipient of the antigen presentation. But the other 3 types are able to produce, are able to present antigens for that handshake to occur. Okay, so moving on to section 13.4, we're going to describe the main differences between T helper cells and T cytotoxic cells, explain how naive T cells become sensitized to an antigen. We've talked about that process a little bit because we've talked about the gene that gets snipped up and pieces thrown out and pieces mixed up and flipped over and all that sort of thing to be able to have the diversity to process virtually any kind of antigen. And note the similarities and differences between that strange type of T cell that's a little bit more innate than the other types of T cells. So it's more of an innate immune response in that it recognizes some of those molecular patterns that the innate immune system also responds to. Okay, so we're now at the stages 3 and 4 of that immune response, that cellular immune response. So T cells are really, the response by T cells are really the most complex and diverse in the entire immune system. And as we mentioned, there are several subsets of T cells, each of which kind of has a slightly different job. Okay, the actions of the T cells are actually dictated by those antigen presenting cells. And that has, in terms of which MHC complex is involved, in terms of which of the antigen presenting cells actually does the presentation, and the cytokine milieu that's around that handshake. All of those things will make a difference in how the cells respond. Okay, so there are restricted interactions that require a particular type of MHC to be recognized before they can be activated. Okay, so you actually have a more specific handshake that has to occur before the activation takes place. Okay, all T cells are going to produce cytokines, and each of those cytokines has a different biological effect. And so again, the kind of soup that the T cell is swimming in in relation to the cytokine milieu is going to make a huge difference in the overall response. Okay, so the end result of T cell stimulation is the mobilization of other T cells, B cells. So again, the helper T cell may wake up a B cell if that's the appropriate immune response. And also, T cells are able to kind of wake up the macrophages. So there's kind of a back and forth there. So one macrophage may have presented a particular antigen, but then the T cell may say, ooh, we need to get more macrophages involved. And it may release a cytokine, which will actually cause more of the macrophages to get involved. Sort of to say, hey, guys, you need to go and eat everything you see that has this antigen involved. Okay, so as we said, there are multiple types of T cells, okay? Let me also introduce the concept of a Th1 versus a Th2 reaction, because depending on what kinds of cytokines are put out there, it's going to decide whether a cytotoxic T cell is needed to kill a particular infection, or if an antibody response is going to be more appropriate. So if you think about it, there are certain pathogens that actually go intracellular to avoid the immune system. Well, if all we had was an antibody response, if the pathogen was inside the cell, the antibody couldn't get in there. And so these cytotoxic T cells are able to take care of that particular kind of situation. So as an example, the parasite that I studied was an intracellular parasite. So once it actually infected that nucleated cell, there's nothing on the outside that would call in any kind of an antibody response. So that one automatically made the Th1 response more appropriate. So everything that was involved in the immune response to that particular parasite was Th1 oriented. Okay, so Th1 tends to have, or Th1 types of helper T cells tend to have CD4 receptors on the outside. And they activate that cell-mediated immunity pathway, okay? So they're the ones that are going to be waking up cytotoxic T cells, okay? And it secretes cytokines like tumor necrosis factor and interferon gamma, okay? Now, this type of T cell is also involved with a type of immune response called delayed hypersensitivity. And we're going to talk about that when we do the lectures for chapter 14. Okay, so it's an allergy that tends to occur hours or days after contact. And these type of T cells also secrete a cytokine called IL-2. The Th2 type of helper T cells are useful to deal with helminth infections as well as protozoans. Okay, so this would be the type of a thing that would be involved in response to amoebic dysentery. Okay, it phagocytoses extracellular antigens. Okay, and it contributes to type 1 allergic hypersensitivity and can encourage tumor development. Okay, so again, you have to be careful. This Th2 also starts off the antibody, the B cell antibody reaction. So it's a good thing as well as it can be a bad thing in the wrong situation. Now, helper T cells with Th17 also has a CD4 on the outside, but its job is actually to increase the inflammatory response. So in some cases, the increase of inflammation is a really vital part of making a successful immune response to a particular pathogen. But Th17 also seems to be involved in some of those inflammatory autoimmune diseases, things like rheumatoid arthritis. So this is what causes that huge inflammatory response around the joints. Okay, so T follicular helper cells have CD4 and CD40, and this is very much involved in the whole antibody response, and in particular, antibody class switching. And we're going to talk a little bit about the different types of antibodies a little bit later. But for example, you start out with one type of antibody during the beginning of an immune response, and then it switches over to a different type, which is perhaps a little more specific. And again, we're going to talk about that in a little bit. Okay, we've also talked about T regulatory cells. Okay, so these are the ones that sort of ramp the immune response down at the end. Okay, and part of that is to prevent autoimmunity. If you have the immune system just going crazy looking for a particular pathogen, it may make a mistake and start, especially if you have a cell that is infected by that particular pathogen. It's possible if you don't ramp that immune response back down that it could flip over into an autoimmune response. But at a minimum, you just want to save all the resources. You don't need to be making a ton of T cells to an infection that's already under control. Okay, so again, it's part of the ramp down at the end of the immune response. And then we have the cytotoxic T cell, and it is characterized by having CD8 on its cell surface, as opposed to CD4, which is the regulatory types of T cells, and the helper T cells, which are kind of the ramp up regulator, as opposed to the T regulatory, which are the ramp down regulators. Okay, and again, these types of T cells are particularly useful for anything with an intracellular infection. Okay, so any kind of foreign cell gets lysed by these cytotoxic T cells. Complex microbes, this is often really an important piece of that. Um, if the immune system can recognize cancer cells, this is the one that goes after them. Okay, viral infected cells, again, viruses go intracellular, so this is the type of cell that would be necessary to control a viral infection. This one is also involved in graft rejection. So again, this is going to recognize a host cell as not self, and it's going to attack it. Okay, this particular type of cell needs MHC1 in order to function properly. That's part of the handshake involved with waking up the cytotoxic T cell. Okay, and then we have these kind of strange bird T cells called gamma delta T cells, and they react both specifically and non-specifically. So this is the one that's kind of sort of innate immune system, sort of adaptive immune system, and it's kind of an odd bird. And again, we're not going to talk a huge amount about it, but it is one of the class of T cells, and so we need to at least mention it. It tends to be specifically responsive to any lipid-based antigen. Okay, so T helper cells basically have that CD4 marker and are critical in regulating the immune reaction to various antigens, particularly as the immune response is building. And they are also involved in activating macrophages. So after that first antigen presentation happens, the T cell, amongst many other things that it's going to do as it goes around and kind of wakes up that whole immune response, is it's going to get macrophages involved. Because again, macrophages are incredibly important for actually taking and digesting those bacteria, so to stop them from being able to reproduce and continue the infection. So it can do that directly by receptor contact, and it can also do it indirectly by releasing cytokines that macrophages respond to. So interferon gamma is an example of something that really wakes up a macrophage. Some of these helper T cells secrete another one called interleukin-2 that basically wakes up, it causes that clonal expansion of T cells. So IL-2, amongst many other things, will wake up that whole clonal response, including those cytotoxic T cells. So if this is a response that requires a cytotoxic T cell, this is how that happens. It secretes some other things. So in the case where a Th2 response is needed, in other words, an antibody response, it's going to secrete IL-4, IL-5, and IL-6, all of which stimulate B cells. And that's going to cause them to do clonal expansion, and it's also going to start the process of secreting the antibodies. And there are a lot of helper T cells. So basically, it's about 65% of the T cell population. And that's because it does so much in terms of the broad range of the immune response. Okay, so here is a picture of T cell activation, and it's basically much of what we've just talked about. So if you look at the top kind of green section, you have a dendritic cell. Remember, it's got those little spiky projections presenting an antigen to a helper T cell, which has CD4. Okay, and one of many things can happen then. That CD4 cell may divide and form a memory cell. It may produce IL-4 and other B cell activators and cause B cells to start clonally expanding and secreting antibodies. It may produce tumor necrosis factor and interferon gamma and start going after cancer cells, for example, or it may wake up the macrophages. So um, and again, like I said, stimulation of macrophages. It may start putting out IL-17, which is inflammatory, or it may uh, sort of turn into a T regulatory cell and start to dampen that immune response at an appropriate time. When the infection is under control, it's going to start the whole process of slowing the whole immune system down through the use of T regulatory cells. Okay. So we've talked a bit about cytotoxic T-cells, and these are cells that can destroy other host cells. So this is the primary mechanism to deal with virally infected cells. Okay, so it will recognize viral peptides on the outside of the cell surface, and it will then go in and attack and lyse that cell before the viral particles are ready to be released, and therefore break that viral life cycle. If it recognizes cancer cells, now part of the problem with cancer cells is that they undifferentiate, and so they don't have as many markers on the outside of their cells, and so it's easy for them to evade immune response. But in some cases, those cancer cells are recognized by the immune system, and the cytotoxic T cells would be the ones that go after that. And of course, any cell from any other animal or any other human that does not have markers that match our self set of markers, the cytotoxic T cells are the ones that go after that foreign tissue. Okay, so gamma delta T cells, again, they're distinct from the other T cells. Now they have T cell receptors, but they're rearranged to kind of be more forgiving in terms of the antigens that they accept. So unlike most T cells, which are highly, highly specific to a particular antigen, this is a little bit more able to accept a broader range. The official term for that is promiscuous. They are promiscuous in terms of their ability to accept those different antigens. And they, again, are frequently responsive to molecular patterns in the same way that the innate immune system looks for those molecular patterns. They respond very quickly, and they produce memory cells. They are particularly responsive to certain types of phospholipids and to certain types of tumor cells with the tumor cell kinds of markers on them. And it's sort of a bridge between innate and specific immunity, although they are not involved in the handshake. That is really the interface between them. But they're kind of neither fish nor fowl. They're not really innate and they're not really specific, adaptive. They're kind of in the middle. Okay, so additional cells that we just kind of need to be familiar with. Natural killer cells are, again, looking for specific antigens. They circulate throughout the body, and these are really the ones that are most likely to attack cancer cells and virus-infected cells. They do it in a fashion very similar to T cells, but they're not T cells. They're not really a part of specific immunity, but they are woken up by certain cytokines produced by T cells, specifically IL-12 and interferon. So it's a different kind of innate or of immune system cell, but again, they're kind of a little bit more in the innate category than in specific, but they're very, very important components of the immune system, particularly for these virally infected cells. And then there are natural killer T cells, not to be confused with just natural killer cells. So these are kind of sort of natural killer-like T cells, but they are T cells, okay? So they have T cell receptors, and they have the ability to release enormous amounts of cytokines and very quickly. And so what that does is it facilitates the other types of cells in actually doing the killing, because it gives it that cytotoxic cytokine milieu that those cytotoxic T cells work in. So again, it's another one that's kind of in between. It's not exactly innate and it's not exactly adaptive, but it's definitely very effective for certain types of infected cells. Okay, so concept check 4. So this is the end of section 13.4. Cytotoxic T cells are programmed to kill all of the following except, again, think about what cytotoxic T cells do. They recognize cells that need, host cells that need to be killed for one of various different reasons, okay? So cytotoxic T cells really do not focus on bacteria at all. They will focus on host cells that are infected by intracellular bacteria, but they're not focused on killing the bacteria themselves, okay? Okay, 13.5. So we're going to look at the steps in B cell activation. So we've just talked all about T cells. So here's the other half of adaptive immunity, including all of the different types of B cells that are involved. We're going to be looking at antibody molecules in great detail to understand how they function, explain the various end results of what happens when an antibody binds to an antigen. We're going to list the 5 different classes of antibodies and important facts about each. So remember, we talked about how there's a process in the middle where the class of antibody switches, class switching. This is where we're going to talk about that. So we're going to draw and label a graph with time on the horizontal axis and the response on the vertical axis that shows the development of primary and secondary immune responses, okay? So it's really important to understand the whole time sequence of an immune response. Okay, so let's start out with B cells. Okay, so we're going to talk about B cell activation. So remember, B cells, by and large, are activated by T helper cells. Okay, so the antigen presentation happens to the T helper cell, and then the T helper cell goes and finds the B cell and does the handshake with them. Okay, and again, some slightly different cell surface molecules are involved, but the handshake is very similar. The purpose is to wake up the B cell to say, hey, you need to go and deal with this. It starts the process of clonal expansion. So the B cell is going to start multiplying very rapidly. And those B cells that are created as daughter cells are going to be plasma cells. So those are the ones that are going to go out into the bloodstream and secrete all of those antibodies. Okay, so there is a regulatory B cell, which is involved in ramping down the B cell reaction. Okay, and again, it's sensitive to a particular cytokine milieu as well. So the normal path is the one that goes directly down from the activated B cell. So it creates a memory B cell, which sort of on the side, it creates a memory B cell to allow future antigen present, or when the same antigen occurs in the body, it's able to produce a massive response, okay? But again, the purpose is to do clonal expansion of these B cells, okay? And so the last one is the creation of these plasma cells, which go out into the bloodstream and produce antibodies and actually go to the site and fight off the infection there. So, antibody structure and function. Okay, so antibodies are Y-shaped. And if you remember the chart that we looked at in the last lecture, you have that constant region in the bottom, that's the bottom of the Y. And then you have that variable region, which is the two arms of the Y. Okay, so there is an antigen binding fragment, which is that Y shape. So the groove in that Y is able to grab onto that epitope, that antigen that is being sought by that particular antibody. Okay, and then there is that constant area, which is that species-specific part. So basically, there are two different protein chains that are made to form this antibody. There's the heavy chain, which is the longer one. And so part of that heavy chain is in the variable area, and part of that heavy chain is in that constant region. Okay? Then there's a pair of light chains, and that's only the variable area. Okay, so that's kind of attached to either side of the Y, formed by the heavy chain. And basically, that region between that constant area and the antibody or variable region, the one that's antigen specific, actually allows that antibody to swivel and attach to a very particular part of that antigen. Okay, so here's a picture, sort of a cartoon, and then over on the right is a little bit closer to the shape that that protein actually takes when it, kind of winds itself up into that tertiary structure. So those green parts are the light chains, and the blue part is the heavy chain. Okay, so as we mentioned, there is the longer heavy chain, so that's kind of the dark magenta, and then that variable region up at the top of it, okay? And then the light chain are the two that are on the outside of the Y, okay? So at the bottom of that constant region is a binding site for cells. So again, that's where binding between, say, the macrophage or anything that is sort of hunting for something covered with an antibody. So macrophages will actually use that heavy chain, that constant region, as a surveillance mechanism. So if you have, say, a bacteria coated with antibodies, the antigen-specific part, that Y, so let's say here's the bacteria, is going to attach to the antigen on the bacterial cell wall, and what's sticking up is that constant region. So this is what the macrophage is looking for, and this is what's attached to the actual bacteria, that Y shape. Okay, so when a macrophage is doing its surveillance and it sees something totally covered with these antibodies, and that constant region is sort of sticking out there, it's going to say, okay, this is something that I need to eat, and it'll go and grab that whole complex and digest it. Okay. I believe that is everything that we need to talk about on this particular slide. Okay, so we have that hypervariable region at the end of the Ys. So it is the actual site on the antibody where that epitope binds, okay? And the amino acid content of that region is extremely varied. That's part of the whole variability of the antibody response. So in order to be able to be specific for a particular antigen, you need to have a lot of variation in that hypervariable region. Okay, and the specificity of that antigen binding site is very similar to the mechanism when we talked about how you have that active region in an enzyme, and it has to do with the actual amino acids that are in that active site. Very similar kind of thing to the antibody. It basically is a chemical match for the makeup of that antigen. Okay. Now, the two FAB sites, the two hypervariable sites, are actually identical. And so you're basically grabbing onto two identical parts of that antigen. It sort of grabs onto it. So it can bind an epitope on the same cell, or on two separate cells and actually link them. So let's say you have an E. coli here that has an antigen here, and you have an E. coli here that has that same antigen space. You can have an antibody come and one arm links this guy and one arm links this guy. So now you're starting to agglutinate all of those E. coli together. You're forming a big blob of E. coli that's all threaded together by all of these different antibodies, okay? And once you get that huge immune complex, that becomes a real target for macrophages in particular that are going to start looking for those kinds of things, okay? Okay, so a summary of antibody function. So the one in the upper left corner where you have the sort of rod-shaped bacteria that's coated with those antibodies. And again, you have the Y facing the antigen on the cell, okay? And then you have that constant region sticking out there, which the macrophages are going to use to recognize that this is something they need to deal with. And so in the middle, you have a picture of a macrophage slurping up all of these antibody-coated bacteria and actually doing the disposition. Okay, now if you have an antibody to a virus, so remember you have that protein coat on the outside of a virus, there are antigens involved with that. So if the virus is coated with antibodies, it can't get to those binding sites that it needs to in order to be able to inject that DNA into the cell. So it physically blocks the virus from being able to do what it needs to do. Now, if you look at the lower right hand, that was what I was just talking about, where you have antibodies cross-linking these bacteria, and it causes it to agglutinate. That is the whole complex, that mess of bacteria and antibodies kind of all in a big blob. Okay, that's referred to as agglutination. And again, this kind of sequesters all those bacteria so they can't do their infection thing, and it really is a big target for macrophages to come and slurp all of that up. The next thing that antibodies can do is basically call the complement system over. So a bacteria that is completely coated with these antibodies will start to bring in pieces of that complement cascade. And once you start the complement, then it will finish it, poke holes in the bacteria, and kill it that way. Okay? And the other thing that antibodies are able to do is essentially coat toxins to make them essentially non-toxic. So again, you have the toxin, you have the antibodies coating it, that gets picked up and dealt with by these macrophages. Okay, I'm going to stop here and we'll pick up in the next lecture.